and welcome to our second BioXTech webinar. We are so fortunate to be able to have Dr. Mohiuddin here today to discuss with us his exciting and groundbreaking work on xenotransplantation. I discussed with Dr. Mohiuddin, and we will run this session as interactively as possible, so we encourage you to ask questions as we go. I will be the moderator. You can either raise your hand, use the raise your hand feature in Zoom, and I will call on you to ask your question, or alternatively, you can type your question into the chat and I will ask your question on your behalf. Now I will kick this off. I am honored to introduce Dr. Mohiuddin. Dr. Mohiuddin received his MD degree from Dow Medical College, Karachi, Pakistan. And after completing his preliminary surgical training at Civil Hospital Karachi, finished his fellowships at the University of Pennsylvania and the Institute of Cellular Therapeutics at Drexel University. Dr. Mohiuddin has been involved in xenotransplantation research since 1992, and he has started xenotransplantation research programs at the National Institutes of Health, the University of Pennsylvania, Philadelphia, and Rush University, Chicago. His contribution of more than 30 years to xenotransplantation includes demonstration of the longest survival of heterotopic and orthotopic cardiac xenotransplantation. Dr. Mohiuddin's persistence in xenotransplantation directed him to accomplish the first human cardiac xenotransplantation at the University of Maryland Medical Center in Baltimore, Maryland on January 7th, 2022. Dr. Mohiuddin, the floor is yours.
Sure. Thank you very much. Thanks for this opportunity. Before I start, I want to know a little bit about your group, your club. I mean, could you give me an introduction of your club?
For sure. So we are a virtual pan-school club that explores the intersection of biology and technology through meeting with experts like you. So I personally was very interested in biology, but I also am super interested in technology. So I like coding, and I took AP Computer Science last year, and I wanted to find a way to sort of learn how to bring these two together and how they can work together, two of my favorite things, and how they can work together in the real world. So that's how we started this club. Yes. Yeah,
no, great. So what do you guys know about xenotransplantation? I mean, what background knowledge do you have about xenotransplantation?
So we actually were assigned an article in class by Mr. Garton, one of my favorite teachers, my AP Bio teacher, Mr. Garton. And we read about the first ever cardiac xenotransplantation that took place, the pig to human heart transplantation. And I was very curious about everything and how it worked. So I decided that this was the perfect opportunity to reach out and maybe learn more about it in the form of this club. Okay,
great. So, I mean, xenotransplantation is transplanting either an organ or a cell from one species to another. So it doesn't have to be human. It could be from a rat to mouse, mouse to hamster, or anything. It could be an organ, it could be a cell, it could be, another tissue. So I'm focused on the heart xenotransplantation. So I'll talk more about heart xenotransplantation. So so why we are doing it, right? First of all, why there's a need for xenotransplantation. So if you look at the number of people who are waiting for an organ transplant, so there is a thing called waiting list, and you are categorized based on your need, how quickly you need an organ for transplantation. So there is about 150,000 people waiting for different organs, mostly kidneys, then heart, and also like liver and lungs and pancreas. So but most of these people, while they are waiting for these organs, will die waiting because there are not enough organs available. Even every one of you or me and everyone here becomes an organ donor, still we will not be able to, make up this difference. So there's a need for something else, right? Another method to save these people's life. So first, you have to understand what is end organ failure, which is there are diseases or conditions where the heart or other organs reach at a point where with medicine or other support, these organs cannot function anymore. They have to be replaced, or in case of kidney, you have an option of dialysis that, your kidneys are not working. You take this machine, hook it up with your vessels and start, taking out all the toxic stuff from your blood and, putting back the needed electrolytes and other stuff in your body. But for heart, there is no such alternative available. When you reach the end stage heart disease, that's the end of your life, unless you are offered a heart transplant. There are a few other alternatives, what we call the assist devices. These are mechanical devices. So they can help one chamber of the heart, contract, mechanically, and they are battery operated. So they are dependent on battery. They are like mechanical things that can fail anytime. There are artificial hearts also. I mean, there are a few that have been tried, the total artificial heart. But the main problem is how to keep the blood circulating within these artificial organs or artificial means, because the blood has the tendency to clot very fast. And none of the devices they have produced have the capability to keep the blood prevent the blood from clotting. And for that matter, the patient has to take anticoagulation for the rest of the life, and at a very high amount, which has its own complications. You get a small bruise, you kind of bleed profusely. So I mean, while there are options being explored where you can grow organs in a petri dish outside with the stem cells, you can. There are options available, and people have even tried to grow human organs in another species. But xenotransplantation is not that. So in xenotransplantation, it started like in early 1600 when a nobleman lost his skull, had an accident, and lost a piece of his skull. So they took a dog's piece of skull and mashed it, and that's how it saved his life. Then in World War I, there were a lot of burned patients. So they take frog skin and replace their burned skin with the frog skin. So there have been many examples of these kind of trials, right? So but the major trial that took place in recent years were Dr. Keith Riensema putting chimpanzee kidneys into several humans, right? So these kidneys lasted for only a few days, and then they got rejected. Then the most famous recent transplant, xenotransplant, is known as baby Fay heart. This was a small baby who needed a heart transplant, and a baboon heart was given to her. But this heart only lasted I think three weeks, and the heart was rejected. So similarly, there were other attempts with what we call non-human primate hearts, which includes baboon, chimpanzees, and other monkeys, right? So then we found out that the baboon heart is not an ideal. The baboon is not, or the other non-human primates are not the ideal source for the organs. One, because they grow very slowly. They are endangered species. There are not many available, right? So and they also have known to transmit diseases like HIV from non-human primate to human, and to get say a heart from me from a baboon, or I have to wait for that baboon to get to my size, which will take about like 20 years, right? So for those reasons, we decided not to use baboons as the donor. So then we looked at other animals, and then we decided about the pig. And why pig? First of all, the pig genome, the genetic material is the entire genome is very well known. So, and with the new technology of modifying that genome, we can add or delete genes, which I'll talk a little bit later about, from that genome. So we can customize that pig, right? Then the pigs are can be bred in captivity. They are easy animals to breed. They grow fast, right? So to get the same heart for myself from a pig, I would only have to wait like 1 year. So the patient that we did recently in January, the pig was only 1 year old. The patient is 57-year-old. So quickly these patients, these pigs become good donors. Then since a pig has been used, is used in most of the world for dietary purpose, right? And there are other uses of pigs, and they are kind of sacrificed for that purpose. So we believe that there will be some less ethical issues with it. So these were some of the reasons why we chose the pig. So what happened next? When we took a pig heart and transplanted into a baboon just to test, right? So baboon was used as a because the baboons are very close to human. So instead of using, you cannot do experiments in humans, so you have to use an animal model. So instead of human, you used a baboon and you transplant a heart in the abdomen of the baboon. So what happened was, within few minutes, that heart, rejected. So, as I mentioned, the baboon heart took like weeks to reject, but this heart rejected within minutes to hours, and it turned black. So we found out that, all the vessels, were clotted somehow. So then, we discovered that millions of years ago, more than one enzymes in our carbohydrate, chain development were kind of knocked out from our system. So that's why some of our blood lining the endothelial cells that line the blood vessels don't have certain sugars on them, and because of that, actually the humans have antibodies that recognize these sugars, which are present on pig endothelial cells. So human don't have it, so they produce these kind of antibodies against these sugars. And pig, where this enzyme was not knocked out, have those sugars. So when you transplant a pig heart in human, these antibodies recognize the pig organ as foreign, attack the cells having those sugars, and a cascade of reaction takes place and causes the blood to clot within those vessels. So this is called like a hyperacute rejection within minutes. So what we can do, we found a way, as you guys remember, probably you guys don't, that the first cloned sheep was Dolly sheep. This was the first time a sheep was cloned, and this was the first time anything was cloned. And the same company that did that started making pigs. The first thing they did was they they kind of knocked out the same gene that we are missing from the pig also. So now the pig don't have that sugar on them. So even if we have antibodies against that sugar, there's nothing to bind and attack. So we found 3 such sugars on the pig. So we deleted all those 3 sugars. Then we start seeing that these hearts or other organs, grow fast even after transplantation. So we kind of knocked out a gene, which is called growth hormone receptor gene, to retard the growth. So these pigs with these gene knockout grow slowly, and their heart after transplantation or organ don't grow out of proportion, right? So those were like 4 genes that we knocked out. Then we found out that there are a lot of mechanisms that keep the blood, prevent blood from clotting, and prevent immune reaction. But there are molecules of the pig that are incompatible with the system of human. So what we did was we identified 6 such molecules, and we inserted those molecules in the pig. So in general term, how this is done, right? So what you do is there's a technique, you must have read about it in your biology class, CRISPR-Cas9. It's like basically scissors. You have a genome, you cut the piece of gene out and put whatever you want in there, or you just cut and take the gene out. So that construct is made. What you do is when the pig is pregnant, you take the eggs out, you take all the materials from the egg out, you suck it out, and you put this new constructed material into the egg. You put the egg back into the ovary of the pig, and after it completes its gestation period, the new pig is born, and that pig has the genetic material that you inserted in the egg. So that way, this is the method that we use. So before we used to do 1 gene or 2 genes at a time, and then crossbreed these pigs to get 4 gene pig, right? So now with the CRISPR-Cas9 technique, you can knock out as many genes as you want, and you can insert as many genes as you want, and then you have what you need. So what happened with this didn't happen in a day, right? It took me like 15 years to get to this place. So in this process, we tested different genes, gene combination, different. So even with 10 gene modification, you still get rejection, not hyperacutely within minutes or days, but in maybe like months. So what we did in a sequential manner, we started testing different gene combinations. And also on the other hand, as you know, the immunosuppressive drugs, the drugs that you give to suppress rejection, like even if you do a human-to-human transplant, any organ, you have to take medicine for the rest of your life to suppress your immunity enough so the heart is not rejected. So that we also, while we were working on the genetic manipulations, we also worked on different kind of drugs that we can use instead of using generalized immune suppression, which doesn't discriminate between any cells and kind of suppress all the cells in the body. We focused on specific pathways and I in particular was more interested in the CD40 ligand pathway. I don't know if you guys have studied that. This is like a signal 2 between the antigen presenting cells and T cells, or B cells and T cells. There's a crosstalk between them. So our idea was to stop that crosstalk between T and B cells, because both of them were responsible for having this immune reaction against the pig cells and causing rejection. So that did work. So even in the abdominal model, where we transplanted heart in the abdomen, while the baboon's original heart was intact, that heart, lived for about 1,000 days, like almost 3 years. And we went, and at that point we thought that we induce some kind of tolerance, which is a mechanism where you can teach the immune system to recognize foreign cells or organs as self, and after they are taught, you don't need any other supportive immunosuppression to keep them, to prevent them from rejection. So we thought that we have reached that stage. We withdrew this main drug that I mentioned, but to our surprise, that all those hearts rejected after 2 months, because we had not actually induced tolerance, and these hearts required their medicine to continue even after 3 years. So that was proof of concept that you can, from minutes of rejection, we can prolong the graft to years. So we thought that, okay, let's test this heart in its original position. So we started transplanting this heart in the baboon and taking out baboon's heart and putting this heart in. So then with a lot of manipulations and changing genes, gene modifications and drugs, we got to a stage that we started consistently surviving the baboon heart in the chest, because when it was functioning in the abdomen, it was beating, but it was not performing the function that heart performs in the chest. So finally, once we reached that stage, then we went to the federal regulatory authorities and say that we have this kind of data that we have shown in the animal model, which they require. Will you allow us to try this in the human model? And they said no. They said you don't have enough data. You should continue to do this. They gave a specification what they wanted to know about, wanted to see in our non-human primate preclinical data. So we are doing all that. But in the meantime, we came across this provision that FDA, the Federal Food and Drug Agency, allows anyone to use, in which if you have a patient in a condition, where you have tried all the conventional means to save life and you are not able to do it, they allow you to, for just for that patient, a procedure or a drug that is in an experimental stage, is not approved, and you can try that and try to save patients' life. So Mr. Bennett came to our hospital 2 months prior to this transplantation. He had heart failure. His heart was not functioning properly, and it was not pushing the blood through his body. So we had to put him on a machine that was acting as the heart. And he was on that machine for 40 days prior to transplantation. Mr. Bennett had poor compliance history. He never went to the doctor, or he didn't take his medicine at the proper time, or so I mean, that is one of the criteria where you are denied of a human heart. So there's a committee in the hospital that looks at your record, and because these human hearts are so few, you have to justify giving it to someone, and you cannot give it to someone who cannot take care of it, right? So the committee decided that this patient is not eligible to receive a human heart. And also we consulted several other hospitals, because every hospital has different criteria. And then we consulted hospitals across the state, and none of them accepted him as a heart transplant patient. So at this stage, we we proposed to him first before even going to FDA that, we have this experimental procedure where we have used a pig heart and it has functioned very well in a baboon. It has supported the baboon's life for almost 9 months. And, would you like to be part of this procedure and accept a pig heart? And he said, yes, of course, because he had no other, if he had not done this, he would have survived for only a few more days. So he was in his full conscious. His mental status was alert, and we had, several psychiatric evaluation done on this patient. So to make sure that this patient is capable of giving his consent, he even said that, if I get this heart, will I be doing oink oink? So that tells you that he was mentally capable. So once we have his verbal, consent, we went to FDA and say that, we have this patient and this is the only way of saving his life. So, and this was December 20th during the holiday period. So throughout the holiday period from 20th to December 31st, we've been going back and forth with the FDA, several meetings, they kept sending us more questions. We started replying. They wanted to see all the data that we had acquired. So finally, on the December 31st, on New Year's Eve, they gave us permission to do this transplant. But then, then we found out, okay, we got the permission from FDA, but who's going to pay for it? This patient may stay in the hospital for 2-3 months, and it will cost maybe 1.5 million or more during that stay. Who's going to pay for it? Because insurance doesn't have this in their manual, this procedure, and and when we approached them, they said, no, we cannot pay for this procedure. So finally, we had to convince our own institution to absorb this cost. Since he was already admitted in the hospital and being taken care of, they finally, after it took them 7 days to give us that permission. In the meantime, we had to draft a very elaborated consent form that included every possible thing that can go wrong with this patient, right? So we have to be very upfront with them. We didn't promise him that, we'll give him a long life. We said that this is an experimental process. Based on our data, we can do this. And we expect you to live a little bit longer. But the one difference here was that this patient was very, very sick. He's been on the bed for 2 months. His muscles were totally wasted, right? He hasn't gone out of his bed. So this was a big challenge compared to the healthy baboons where we were used to putting the heart in, right? So this was a challenge. But he consented to it. And so on 7th, we transplanted this heart in this patient. And since then, so far he's doing very well. In a sense that his heart is not rejected. It's been today's 46 days post-transplant. And the heart has not shown any signs of slowing down or rejection. We've been carefully monitoring. He has a celebrity status in the hospital, right? And everybody wants to take care. Every doctor wants to take care of this patient. We call one cardiologist, we get 5 coming in to take a look at him. So, is he still in the hospital? Yes, he's still in the hospital, and he'll be in the hospital for extended period of time, because the issue we are facing now is to get him back to the same strength that he was several months before he was admitted to because his muscles are totally wasted, right? So although the heart is working fine, I give the example that we put a Ferrari engine in a 1960 car, right? So the heart is the function fine, but his body needs to cope with it. So, and then, the one major thing in transplantation is to create a balance between immunosuppression and prevention of infection. So if you immunosuppress this patient more than you need to, there's a chance of him getting infected, right? So you are suppressing his defense that can go against him also, and this is happening also. I mean, he's getting off and on small infections that delaying his recovery. So he's now getting physiotherapy every day. A physiotherapist 3 times a day comes in and tries to do different exercises, try to bring strength in him. I mean, there are we are providing nutritional help. A lot of things are being done to him. We are constantly checking because this is the first, right? So every day we are learning a new thing. Nobody has done this before, so we don't know what will happen next. So every day, based on our baboon data, we are anticipating what could happen next, and we are trying to kind of counter it beforehand. The biggest problem so far we have faced is because of infection. There are some infections that typical for immunocompromised patients like him. So I mean, definitely he's getting those infections, but luckily we've been able to treat those infections. Otherwise, his progress has been. We were expecting like by 2 months we'd be able to send him to a rehab facility where he'll go through different rehabilitation maneuvers, and then after a month after that, so we could discharge him. But this has not happened. His progress has been a little slower. He's not regained enough strength to be discharged to the rehab. So it may take. But everything is moving in the right direction. It's slowly gaining the strength, and hopefully in a month he'll be into the rehab, and then maybe a month of rehab he could go to his home.
Yeah.
His only wish is to go home and walk his dog. So so one very important thing in transplantation is the motivation of the patient. In any kind of surgery, you have to be the patient has to be very motivated. He could have given up, a day after surgery and said that, okay, no, I don't want to live, right? But this particular patient has a will to live, and he's constantly fighting everything, and and I'm sure that he's gonna survive.
So how can you guarantee, due to his past history, how are you sure that his will to live is truly strong due to his past history of not taking his medicine, not going to doctor's visits?
Because he didn't understand before that, he was not taking his medicine to harm himself. He just was lazy, right? I mean, he just didn't want to go to the doctor, and people don't recognize what harm it's doing. Like high blood pressure, simple high blood pressure can kill you, after some time if it's not attended, right? So this patient had that kind of high blood pressure, which he never took care of. He initially went to doctors and they gave him medicine. He never took it. So I mean, but now, I mean, we have taken upon ourselves to follow him. Even when he's discharged, we make sure that he takes his medicine, because in transplant you have to take these medicine, as long as you live. Because if you stop taking this medicine, your heart will reject, and then you can die.
So there are a few questions coming in.
Yeah, and thank you, Elise. Let's, if you wouldn't mind taking the ones like maybe at the bottom there from Samantha that are really related to what we're talking about there, and then maybe you could back up. And guys, I'd like to thank everybody for coming, and thank you for giving us your time to speak to us today. It really means so much to all of us. The teacher in me is stepping in. Sorry, I can't help myself. So guys, there's a variety of different levels that we all have and bring. So no question is too small, no question is too big. So please, please, please feel free to ask anything that you want. Don't feel like any question is like, oh, that's a silly question. Don't feel like that, please.
Okay, perfect. So Samantha's question is, are his symptoms from immunosuppression any more severe than those of a normal human heart transplant?
Of course, you know, when you are, I mean, compared to normal heart transplant. So even when you get a human heart transplant, you are at a stage where some of the other organs are also not functioning very well, and you're not a healthy person. So in this case, it was a step further. This person, you know, was kept alive on this machine for 40 days, and you know, and as long as we would have kept him alive on this machine, he would have lived. So he was a debilitated, and you know, he was very, very weak. So we had some extra challenges in this patient because of his weakness, because not only his physical weakness, but since we are giving him all these drugs, you know, his immune system is very weak. So a small infection that we can fight very easily, you know, he can get very sick with that. So we have to protect him from that kind of infection also. So compared to the normal human to human transplant, this was a little bit more difficult case because of the patient's prior condition, not because of the procedure itself.
Okay, awesome. Another question is, theoretically, could xenotransplantation help lengthen the human lifespan? For example, postponing the time of natural death. If so, are you looking forward to this? And when would this be possible given the current rate of biotechnological advancement?
Yeah, so I mean, you know, that's the goal. That's the big goal that I have. Where, you know, my dream is that no one should die because of you know, non-availability of this of an organ for him, right? So if this works, and you know, there's still a lot to be learned from this, and if we have this perfect system and the FDA allows the clinical trial, and after that approve this procedure, then you know, there will be unlimited supply of organs, right? From one pig you can take heart, lung, liver, kidney, and you know, help 5-6 patients from just one pig. And you know, and anyone who has any kind of this terminal disease can benefit from this. So when a person reaches the stage where he gets the heart transplant, he's already very sick. So I mean, you know, if we have unlimited supply of organ, we can intervene earlier when his other organs have not taken the hit, right? And we can give him that organ and save you know, and prolong the life even further. So I'm very hopeful that this will you know, help a lot of patients, and that's been my lifetime goal.
Thank you for your answer. Marco.
Hey, sorry, I don't want to, you know, chime in or anything, but I just really have a, you know, kind of yearning question here. So sorry again.
Please go right ahead.
You muted yourself.
Yes, you're muted.
Oh, sorry, my fault. Back on what you said about when someone is in a critical condition where they need a heart transplant, how are you, you know, thinking about for the future, for example, right, cancer patients, right? When their cells divide, it's either maybe hereditary or, you know, maybe just something is wrong with their immune. When you put them under an immune suppressant, right? How are you thinking long-term about how, you know, this pig heart might be affected by cancerous cells? Like, have you, you know, tried to or done any tests regarding cancer cells or, you know, with those immune suppressants, or are you still just, you know, theorizing at this point?
No, I mean, so you have a very valid point that, you know, that even this heart that we are transplanting could become infected from the cancer cells. And, you know, if a person has cancer, you can you know, have the cancer cells go to this heart also. The immunosuppression that's used for cancer, different cancers, is a little bit different than the immunosuppression used for heart, and especially the ones that we have used. We have not used the immunosuppression that kind of suppresses every single cell in the body. So we are specifically targeting cells that will cause the immune system to reject this heart. In cancer, also there's a wide variety of immunosuppression that you can use, and but our goal is not at this time to prevent cancer. So I mean, if the patient develops some kind of cancer, it will be treated accordingly based on the cancer it develops, right? But in the heart transplant, there are specific 3 or 4 main drugs that are being used, and those are not typically used for cancer treatment.
Awesome. So Marco has asked, what, if any, pushback have you gotten from people who say the use of heart transplantation is unethical?
I mean, so they have their opinion, and everybody has a right to give their opinion. And we have received a lot of these kind of pushback, and why we need to kill an animal to save a human life, right? But we are doing that for other purpose also, right? For food and other. We wear leather jackets, we use cosmetics, everything. So all those, are part of, have components that come from an animal. So, I mean, this to me, I mean, is a noble cause. I mean, this is what I became a doctor for, to save life, right? So, and this is how, this is one of the methods that we have developed to save human life. I mean, we have taken organs, not organs, but like valves from pigs before to repair or replace the human heart valves. We've used insulin from pigs also, and other small parts to transplant before also. So I mean, they're of course an ethical issue, but definitely saving human life. We are trying to utilize every means that we have available.
Thank you. Hamada is interested in, how does the surgery work exactly? Do they just do stitches around the arteries and veins? And if so, then what is the healing process? It seems like there could be a lot of possible bleeding.
Yeah, sure. I mean, bleeding, any surgery you do, the biggest complication is bleeding, right? So the better the surgeon is, the less is bleeding, right? So this, the heart, as you know, has 4 chambers and have vessels coming out of it, right? I wish I had a model here I could show you. So we cut out the heart from the body, right? When we cut it out, we cut the 2 major vessels, the aorta and pulmonary artery, and also all the pulmonary veins that are going to the lungs. So when we take out the heart, then we sew up the pulmonary vessels. So there are 2 big vessels sticking out, the aorta and pulmonary artery. Then there are 2 chambers, the atria, right? We cut open those chambers. So now there's like a C cup, 2 C cups on the heart, and 2 vessels that we need to attach to the other side. So when we take out the recipient heart, the same way, we leave like 2 C cups there in the atria and the 2 vessels there, major vessels. So we sew them together, like joining it's like a plumbing mechanism. You just join 2 tubes together like this, and then sew it around it, and make sure after you've done that, no bleeding happens. While you are doing the transplant, you stop the blood supply to the heart, and the patient is put on a heart-lung machine where your blood is circulating through a machine and it's acting like a pump, the same as your heart. Once the anastomosis is done, you take out you start the flow back in the heart. Heart starts to pump again, and then you take out the tubing that connects to the machine. So this is like a standard process of a heart transplantation, irrespective of whether you are doing a xenotransplant or allotransplant, which is human to human.
Thank you. Phoebe has asked, when do you believe, if ever, xenotransplantation of the heart will become a common medical procedure?
Yeah, that's a great question. We don't know. So like I said, FDA has given us only permission to treat Mr. Bennett, our patient. We may ask for another patient, but I don't know how many patients FDA will allow us before we go through the normal process of, doing these non-human primate experiments and then doing clinical trials, different stages of clinical trial, and finally getting the approval. The main thing all the regulatory agencies are look for or are afraid of is the safety, right? So any drug or any new procedure, they want us to prove that it is safe for the patient, right? And it also, in this case, they want to make sure that there is no communicable disease that will be transmitted from the pig to human, right? So all those things we have to prove in an animal model first, then in a small scale clinical trial, which will expand in different stages. And finally, when we are satisfied, or the FDA is satisfied, they will give us the approval to expand this to everyone. And this may take like still a few years, a couple of years at least. It all depends on how fast we can move, how fast FDA can allow us to do this. But it's definitely if you had asked me this 5 years ago, I would have said maybe 10 years, or I don't know. So there's a famous saying going in xenotransplantation, that xenotransplantation is the future and always will be, right? So nobody believed in it, right? And somebody said, xenotransplantation is just around the corner, and a very long corner, right? So all these skeptic quotes have been around, but we have proved them all wrong by doing this experiment, and and the patient is doing well for 45 days so far. So I mean, that anything is possible. I mean, you just have to be persistent.
Thank you. Is there anyone else who would like to verbally ask their burning questions? All right. Yes.
I was wondering, so you mentioned that the heart that was transplanted into Mr. Bennett had a couple, meaning he would be having to take medicine for the rest of his life. I was just wondering, what would you do maybe the next time an opportunity like this comes around where you could do a heart xenotransplantation? What would you change to maybe decrease the amount of drugs that it'll take for the rest of their life, or completely eliminate that entirely?
So that is an ideal situation for a transplant surgeon or transplant immunologist to develop a plan or somehow induce this kind of a condition where you never have to take any drug, right? So if we are able to modify the genes of the pig in a way that if we find out that every single culprit, and then we knock out or modify in the pig, so that when you transplant that pig heart or organ without any immunosuppression and no rejection happen, that's in a very ideal condition, and that can very well happen. Either that, or you teach the immune system of your body, of the human body, to recognize pig organ as self, right? When you probably have learned this in your biology class, that through thymus, which is like a university, it teaches the cells, developing cells, to recognize what is its own body, so the cells don't attack your own cells, and then recognize the foreign cells as foreign. So like bacteria and viruses, when they come in, they attack and kill them. So there are ways where and we have worked on those ways also in small animal models, that you can induce this kind of education and make the organ accept without giving any drugs. But in the large animal model, and also in patients who are usually very sick when they are in need of a transplant, this has not been has not been achieved yet, and people are still trying. So some of those two mechanisms, like tolerance or genetic modifications, may one day becomes available, and you will never have to take medicine. That will be the best thing that could happen in transplantation.
Thank you. Sajid has asked, how did the surgeon go about connecting the cardiac nerves and vagus nerve between the human host and pig heart implant? Was there a period of time needed for Mr. Bennett's body to properly train the new nervous system pathways?
Yeah, so this is a great question, because when you do the heart transplant, the heart is de-innervated, means that we never connect the nerves back. So the heart, as you know, that the catecholamines either are secreted from the nerve endings, or they are in the circulation. So the heart now, the transplanted heart, now relies on everything that is coming to it in the blood, not through the nerves. So all brain controls it, but it controls it through the secretion of hormones that are transported to the heart in the blood. So the heart is completely de-innervated. That means that all the nerves are cut; they are never joined together, but still the heart functions, very well.
Thank you. Another question is, it seems like this xenotransplantation is very expensive. Do you think that if this option becomes available to more and more patients, it will become affordable, or will it continue to cost around the same of about $1 million?
Yeah, no, it will not be. So first of all, if it's FDA approved, most of these insurance companies will be able to cover this. And also these days the major cost is also the making of the pig. I explained to you the procedure that takes a long time, and a lot of cost, and to keep these pigs, which are very clean, kept in a very clean facility. These are not the pigs that you see outside in the barn. They are kept in each pig has its own room, and very clean room with HEPA filter and all that. So all that requires money, right? So but when this becomes a routine, they will be produced in a bulk, and in a larger facility and all that. So the cost will definitely come down. On the human side, the cost will be covered by the insurance, and it will not be more than what now human to human transplant cost, right? Which is also a huge cost, but it's covered by the insurance at this time.
Thank you. So one last question before we finish off for the session. We're a bunch of high school students, right? So, and we're in the middle of sort of deciding our careers and learning about what we might be interested in the future. Can you share with us your journey and maybe why you picked xenotransplantation as the main thing that you focus on?
Yeah, so I mean, that's very important that I came to this country, to become a cardiac surgeon, right? So when I came in, I was doing my training for cardiac surgery, and one day and this has been published in several news articles also, because I tell this story to everyone. He sat me down and said that, let's calculate how many people you're going to save, during this, once you become a cardiac surgeon and in your career. So we calculated a certain number. Then he said that, what if you work in a field that may, if it works, it may save, exponentially, maybe several hundred folds of life. So, and that was xenotransplantation that my first mentor was pitching to me. And at that time, it was in its infancy. Nobody knew about it. And, it appeared as a very wild idea, right? But all these ideas sometimes I felt during my career that I made a mistake, because, success was not always there. That I had a very bad experiences. All these research and advancement requires a lot of money, right? Even a baboon transplant that we do is about 500,000 each, right? So somebody has to pay for it, right? So in research, the funding it's very difficult to get that kind of funding. So there have been dry periods, and there have been good periods. Sometimes you go few steps forward and have to take a step backward. But the persistence is the key, right? I always believed in that. I'd be able to get to this point. Although there were times when even my family said that you picked a wrong field. I mean, it's going nowhere. But you have to believe in yourself that whatever you do, I mean, there's no profession in the world that is, that is bad. I mean, physicians are good. I mean, that's a great profession. But any profession you choose, just stick to what you believe in, and and keep working hard on it. And one day you will succeed. I mean, I guarantee you.
Okay, thank you so much, Dr. Mohiuddin, for coming to spend time with us. This was such a wonderful opportunity to learn about xenotransplantation. And I can now see where what we're learning in school can end up benefiting the world. So that's really cool. I'm sure you've inspired many people today, myself certainly included. Dr. Mohiuddin, do you have any concluding remarks for us?
No, just, thank you for having me, and, just work hard, and, and believe in yourself. Anything is possible, that I'll give you another example, quick example. When I was pre-internet stage, when we used to have this rewritable CD or the CD disc, I was holding one CD in my hand, and I was thinking, I asked my fellow resident that, could be a time when we can, instead of using multiple CDs, could we rewrite them? He said, what kind of a fool you are? I mean, these are, they're made with special, the techniques, and you can never rewrite them. And, you guys now know that, after a few years, we had a rewritable CD drive, and now they're non-existent, and probably you guys don't even know about CD drive, probably, right? So, so the nothing is impossible, that if think big, always think big, and try to see, I mean, all these people who've made a lot of money also are not made a lot of money. I mean, they started from somewhere, and they're just because they thought, big, they are at the stage that they are now. So I mean, just don't keep your imagination run wild, and think of new things. You guys are, I understand, at your stage, you are much more brighter than I was. You have more, options available, opportunities available, avail them.
Awesome. Thank you again so much. And for everyone's participation, we host webinars monthly, and there will be many more exciting topics to come. So I look forward to seeing you all again next month. This concludes